Picture this. You've just opened a scientific research paper, right? You are staring at the screen, and what do you actually see?
Like dense columns of tiny, stilted text.
Exactly. Microscopic graphs with axes you can barely read.
And a sudden jarring block of complex statistical equations just out of nowhere.
Right. Right on cue, you feel it. The dreaded brain freeze, the sheer wall of information hits you, and you immediately feel, completely overwhelmed.
Well, it's a remarkably universal experience.
Yeah.
I mean, you look at that wall of text, and the instinctive reaction is just a very quiet panic. You think, I have absolutely no idea what I'm looking at.
And the unfortunate thing is, so many of us internalize that feeling as a personal failure. Like, take a student we were reading about, Selena, out at the University of British Columbia.
Oh, yeah. Her story is so relatable.
Right. She hit this exact wall. When she first started university, opening an academic paper felt practically impossible to her. She'd see those dense paragraphs, and she genuinely believed she was supposed to understand every single concept on the very first read.
Which is, frankly, an impossible standard.
Completely.
But when you inevitably don't understand it all on that first pass, what happens? You start avoiding the primary literature altogether. You rely on, like, summaries or secondhand interpretations.
Which defeats the purpose of the research.
Exactly. And here is the critical problem identified by Professor S. Keyshot from the University of Waterloo. He points out that efficiently reading a paper is an absolutely vital professional skill, and yet it is almost never formally taught.
You were just expected to magically know how to decode it, which really begs the question, why is it so hard to decode in the first place? Like, why do authors write like this?
Well, it comes down to a mix of things. Academic tradition, strict page limits in journals, and this intense pressure to sound rigorously objective.
Right, they can't just be casual.
No, a scientist rarely wants to write a simple sentence like, "the mice died." Instead, they write, "a cessation of biological functions was observed in the marine subjects."
Which is technically precise, I guess, but it completely exhausts the reader.
It does.
And because students and professionals are left to figure this out by trial and error, it just leads to hundreds of hours wasted. The authors of a famous PLOS guide on reading scientific articles, they point out that navigating these papers is fundamentally different from reading a news story or a novel.
Or even a textbook, yeah. If you try to read an academic paper cover to cover, you're going to burn out.
And that is exactly the mission of our deep dive today. We want to rescue you from that trial and error frustration. We're going to provide a complete masterclass on navigating these academic beasts strategically, critically, and most importantly, without the stress.
Sounds like a solid plan.
Okay, let's unpack this. Because before we even look at that dense text, we need a game plan. We have to know why we are looking at the paper in the first place. That's rule 1 from the PLOS guide. You have to lock in on your actual intent.
Right, intent is everything.
Yeah, like, are you just skimming this to see if it's vaguely relevant to a project, or are you doing a comprehensive review? Or maybe you're actively trying to replicate their specific laboratory method.
And your intent totally dictates your entire strategy. But that intent has to be mapped against the author's objective, too. That's rule 2.
How so?
Well, you need to identify what kind of article you're dealing with to set your expectations. Is it primary research, introducing brand new data? Or is it a review article that's just synthesizing a whole field?
Or a commentary piece taking a stand on some controversy.
Exactly, because if you approach a high-level commentary piece expecting step-by-step lab methods, you're going to be completely lost.
Okay, that makes sense. But beyond aligning your intent with the format, there is a vital, non-negotiable step before you read a single word of the research.
Oh, the credibility check.
Yes. You cannot blindly trust a paper just because it has a polished PDF layout or, uses intimidating vocabulary.
No, absolutely not. I can easily see how someone could spend 4 hours dissecting a paper only to realize the underlying science is fundamentally flawed.
Or completely biased, so how do we vet it?
Dr. Jennifer Raff. She's a geneticist who wrote a really highly regarded guide on this. She warns that you should always investigate the source first.
Like checking the authors.
The authors, their institutional affiliations, and the journal itself. You want to ensure the journal is reputable, ideally indexed in major databases like PubMed or Web of Science.
Now, for someone who isn't steeped in the academic publishing world, what do those databases actually do? Are they just, like, specialized search engines?
Think of PubMed or Web of Science more like the bouncers at a very exclusive, rigorous club.
Okay, I like that.
Yeah, they aren't just hosting PDFs for anyone who wants to upload one. They index journals that meet strict peer review and editorial standards. So if a journal isn't getting past those bouncers, you should be highly suspicious of the science inside.
That makes a lot of sense. And this brings up a massive flashing red flag that we all have to watch out for.
Funding.
Yes, funding. You have to scroll down and check who paid for the study. If you are reading a paper on the environmental safety of an agricultural chemical, and you see it was funded by the company that manufactures that exact chemical.
Yeah, you are dealing with agenda-motivated research. It demands a significantly higher level of scrutiny.
Because the science might be sound, but the potential for bias is enormous.
Enormous. Or consider the famous data fabrication lawsuits at Duke University some years ago.
Oh, right, the Duke lawsuits.
If an institution or a specific lab has a history of pending lawsuits for fabricating data, you need to know that context before you invest your cognitive energy in their findings.
So true. Okay, so we've vetted the source, we know our goal, we understand the format, and we trust the paper isn't completely bogus. How do we actually read it? Because, as we established, treating it like a normal book is a recipe for disaster.
Right. Researchers Pruginen and Hewitt from Rice University state this perfectly. They argue that reading linearly, starting at the title, reading through the introduction, all the way down to the citations is the absolute worst approach.
The worst. So what is the alternative? How do we navigate it?
You use what they call the nonlinear strategic hopping method. It is all about jumping around the document with a distinct purpose.
Strategic hopping. Okay, like what?
Well, for example, Helen March, a student from the University of Bristol, has a fascinating hack for this. She suggests reading the conclusion immediately after reading the introduction.
Wait, immediately after, which feels completely counterintuitive.
It really does.
That's like skipping to the final chapter of a mystery novel to see who the killer is before you even meet the detectives.
It does feel like cheating. But in science, you aren't reading for suspense. You want to know the ending right away. By reading the conclusion right after the introduction, you find the author's final destination before you map out the complex journey they took to get there.
Oh, I see. It gives your brain an anchor.
Exactly. It helps you interrogate their intermediate points far more effectively.
Okay, I like that. But here's where it gets really interesting. When people talk about jumping around a paper, almost everyone assumes you start with the abstract, right? That little 200-word summary at the very top.
That is the usual advice, yeah.
But Dr. Raff has a highly controversial rule in her guide. She insists you should read the introduction first and completely skip the abstract.
It is a controversial take, but her reasoning exposes a lot about how our brains process information.
But wait, skip the abstract entirely. I have to push back on this. Isn't that like trying to build IKEA furniture without looking at the picture on the front of the box?
That's a good way to look at it.
Right. You're just staring at a pile of wooden clanks and screws with no idea what the final dresser is supposed to look like.
It's a great analogy, but what's fascinating here is where that analogy breaks down. Dr. Raff argues that the abstract isn't just an objective picture of the finished furniture. It's a highly condensed, carefully curated summary written by the authors themselves.
So it's biased.
It is inherently subjective.
Yeah.
If you read it first, you risk becoming inadvertently anchored by the author's own interpretations of their results. You start seeing the data the way they want you to see it, rather than what the data actually says.
So by skipping the abstract, you avoid getting spoon-fed their narrative. You're forcing yourself to look at the raw materials first.
Precisely. Skipping the abstract forces you to be an active investigator rather than a passive consumer. You read the core sections with an open mind, look at the evidence, and then you only read the abstract at the very end.
That way, you can see if their tidy, perfect little summary actually matches the messy reality of what you discovered in the body of the paper. That makes perfect sense. But being an active investigator requires a framework, doesn't it? If we just randomly hop around without a system, we're going to drown in all those planks and screws.
We definitely will. And this is where S. Keshav's legendary three-pass approach becomes incredibly useful. It is a highly systemic way to digest dense material without suffering cognitive overload.
Okay, walk us through the passes.
The first pass is designed to be incredibly quick, just 5 to 10 minutes. This is your bird's eye view. You read the title, the introduction, the section headings, and then jump straight to the conclusion.
And the goal of this first pass is to answer the 5 C's, right? Category, context, correctness, contributions, and clarity.
You got it.
But hang on, how do you realistically judge the correctness of a complex scientific paper in 5 minutes? You haven't even looked at the data yet.
Well, you aren't checking their math at this stage. You are checking their architectural logic.
Architectural logic.
Yeah. Does the core claim they make in the introduction logically track with the heading of their conclusion? If they promise to prove one specific mechanism in the intro, but their conclusion is about an entirely different phenomenon, the fundamental architecture is flawed.
Ah, so you can spot a logical disconnect very quickly.
Right, and based on those 5 Cs, you make a crucial decision: is this paper even worth more of my time?
And if yes, you move to the second pass, which takes up to an hour, according to the framework. Your goal here is to grasp the paper's actual content, but, and this is a huge relief, you actively ignore the heavy mathematical proofs or hyper-detailed technicalities.
Yes, you gloss right over the dense equations. Instead, you focus heavily on the visuals. You look carefully at the figures, the diagrams, and especially the graphs. You check if the axes are labeled properly, and you look for error bars to evaluate the data's reliability.
Let's pause on error bars, because I feel like that is a visual everyone recognizes from high school science, but very few people fully grasp what is an error bar actually telling me.
Good question. Think of an error bar as a visual representation of uncertainty, or a margin of error.
Okay.
If you were looking at a bar graph comparing the effectiveness of two different medications, and the little error lines extending from the top of those bars overlap heavily with each other, it means the difference in the data might just be random noise.
Oh, wow. So it's a quick visual check on how reliable and distinct their findings actually are without needing to run the statistical math yourself.
Exactly. If they pass that visual check, you should be able to summarize the main thrust of the paper. And for most people, that second pass is enough.
But if you are doing a deep dive for your own research, you have to hit the third pass. And this can take up to 4 or 5 hours for beginners.
The third pass is demanding. This is where you attempt to virtually re-implement the paper.
Wait, mentally recreate their work? How does a beginner possibly do that?
It sounds intimidating, but it's fundamentally a thought experiment. You put the paper down, look at their core hypothesis, and ask yourself, if I had their resources, how would I design an experiment to test this?
So you build the methodology in your head.
Yeah. Then you compare your imaginary design to their actual test. This exercise brilliantly highlights the implicit assumptions the authors made that you wouldn't have. It helps you spot hidden failings or potential biases in their setup.
That is so smart. You know, Professor David Stuckler adapted this concept into his own framework, the bird's eye, the swoop, and the street view. And in doing so, he brings up a harsh truth that I think every reader needs to hear.
What's that?
Not all papers are written well. In fact, many are terrible.
It is so true.
If you are struggling on that second or third pass, it is very often the author's overly complicated, unreadable language causing the friction, not a lack of intelligence on your part.
Recognizing that changes the entire emotional dynamic of learning. You stop blaming yourself for someone else's poorly communicated idea.
Right, and to combat that friction, Stuckler recommends taking strategic breaks to avoid cognitive fatigue. Plus, he introduces a classic technique to test your actual comprehension, the rubber duck method.
Which sounds completely absurd, but it works so well.
You literally explain the concepts of the paper out loud to an inanimate object like a rubber duck sitting on your desk or to a patient friend.
The reason it works is that human beings are incredibly good at tricking themselves. When you read silently, your eyes can skim over complex paragraphs while your brain goes on autopilot, and you convince yourself you understood it.
Oh, I've definitely done that.
Right.
Right.
But when you are forced to synthesize those concepts into spoken words to explain it to a rubber duck, you instantly hit a wall where your understanding actually stops. It exposes the gaps in your knowledge immediately.
So we have our passes, we've talked to the rubber duck. Let's apply this framework to the deepest level of interrogation, what Stuckler calls the street view. If we are tearing the engine apart, what tools are we using on the specific parts?
Well, following Dr. Raff's guide, when you open the introduction, you don't just read passively. You are hunting for 2 specific things. First, you identify the big overarching question. Not just what is this paper about, but what is the giant macro-level problem this entire scientific field is trying to solve.
It's exactly like using Google Maps. You start zoomed all the way out. You're looking at the whole country. That's the big question.
Right.
Then you zoom into the state, the city, the neighborhood. And finally, you find the highly specific question the authors are asking in this exact study. That's the street address.
I love that mapping analogy. Once you know their specific street address, you look at how they plan to get there, which takes you into the methods section.
Which is easily the scariest part of any paper. It is a dense, impenetrable forest of jargon, chemical abbreviations, and proprietary equipment names.
It can absolutely overwhelm your working memory, but Dr. Raff has a fantastic visual hack to combat this. She says, stop reading and start drawing. Literally draw a diagram for each experiment.
Just sketch out what they are doing on a piece of paper.
Yes. Visually map out their physical setup, write down the sample sizes, draw arrows for the sequence of steps.
Mhm.
By translating dense, jargon-heavy text into a simple spatial flowchart, you engage a completely different part of your brain.
That makes total sense. It bypasses the jargon fatigue and allows you to grasp the physical reality of what they actually did in the lab.
Exactly. And once you understand what they did, you hit the results section to see what happened. This is where you have to look at the crucial details. Check every single figure and table. Pay attention to labels.
And look closely at that sample size you just drew in your diagram, right? Was this medical study done on 10 people or 10,000 people? Because that completely changes the weight of the findings.
Absolutely. You also need to watch for highly precise language. Words like significant or non-significant are thrown around casually in everyday speech.
Right. In normal conversation, if I say something is significant, I just mean it's important or large.
But in a scientific results section, significant strictly refers to mathematical probability. It means the result is statistically unlikely to have occurred by pure chance.
Okay, so it doesn't necessarily mean it's a huge deal.
Not at all. You can have a finding that is statistically significant, but the actual effect is so microscopic that it is completely meaningless in practical, everyday application.
Wow. Okay, so I have my sketches, I see the sample size, I recognize the statistically significant data, so what does this all mean?
Well, here's a vital rule for reading the results section. Just write down what the raw data is, not what the data means.
Wait, why wouldn't I want to know what it means right away? Isn't that the whole point?
It comes back to maintaining that active investigator mindset. Avoid interpreting the broader implications at this stage. Formulate your own thoughts, strictly based on the raw numbers and graphs.
Oh, I see.
Yeah, ask yourself, do these results actually answer the specific question they asked? What do you think this data implies?
Hmm?
You want to form your own independent interpretation before you open the discussion and conclusion sections.
Because if I read their interpretation first, I might just blindly agree with it. By forming my own conclusion, I can compare my thoughts with theirs. Did they identify their own weaknesses? Did they overstate their findings? Or did they completely miss a variable that I caught?
Exactly. That process transforms how you interact with information.
Mhm.
But it also requires a highly critical eye.
Which brings us to the human element of all this. Because while we are aggressively interrogating the data, we must also interrogate ourselves as readers.
This raises an important question. How objective are we actually being when we read these papers?
Right. Dr. Michael Mitzenmacher from Harvard notes that reading critically absolutely means being suspicious. You must question every assumption and every logical step. But we also have to watch out for our own brains playing tricks on us.
We really have to actively combat our own cognitive biases. In psychology, there's a concept called response expectancy theory.
Which is what?
It's essentially a self-fulfilling prophecy where humans often find exactly what they are primed to look for.
Oh, is that like when a friend tells you a new movie is absolutely terrible? So you sit in the theater only noticing the minor plot holes, completely missing the incredible cinematography and acting.
That is the exact mechanism. You prime your brain to find what you expect to find. When reading research, you have to ask yourself, am I finding this paper compelling and logically sound just because it perfectly agrees with my pre-existing political or scientific bias?
Or conversely, am I unfairly nitpicking the methods and dismissing it simply because it contradicts what I want to be true?
Exactly. That level of self-awareness is incredibly tough to maintain, but it's so necessary.
It is. But here is the beautiful balance to all that critical suspicion. Rule 7 of the PLS guide is simply be kind.
Empathy is just as important as critical analysis.
Authors are human beings working under immense pressure. Don't let minor typos, a slightly awkward phrasing, or a missing citation completely cloud your judgment of the underlying scientific quality.
Yeah, we have to give them some grace.
And if you are ever in a position to share feedback or peer review, keep it constructive. Our sources remind us that aggressively rude critiques can be absolutely heartbreaking, especially for young scientists who are just trying to find their footing in a highly competitive field.
That empathy extends to how we view the scientific endeavor as a whole, which brings us to Rule 10 of the PLOS guide. Build on it.
Okay, what does that mean in practice?
Think of the entirety of scientific literature like a massive, endless wall of Lego bricks.
Oh, I love this analogy.
Right. Every single paper you read, no matter how groundbreaking, is just one labeled Lego brick. No single paper builds the entire wall of truth.
But by critically analyzing and clicking those individual bricks together, integrating knowledge from multiple disciplines and sources, you build a much stronger, taller structure of understanding.
Precisely.
And as you build that wall, remember the advice from Selena, the student at UBC. It is completely 100% okay to accept that you won't know everything on the first read.
It's expected, honestly.
It is okay to skim highly technical subsections that aren't relevant to you. It is okay to look up foundational jargon in the Oxford English Dictionary. Research papers are written by specialized experts for other specialized experts. Struggling with them isn't a sign of intellectual failure. It's just a normal, necessary part of the learning process.
It's cognitive weightlifting. It's supposed to feel heavy. If it feels too easy, you probably aren't challenging your own assumptions enough.
So let's briefly recap this toolkit we've built for you today. Remember to approach the reading non-linearly. Do not treat it like a novel.
Vet the source and check for those funding red flags before you invest your time.
When you hit the dense method section, literally draw out diagrams so your brain can process the experiment spatially.
Unpack the figures and raw data first, and make sure you form your own interpretations before you read the author's final conclusion.
It is undoubtedly a lot of upfront effort, but it pays off exponentially over time. As researcher Carey and his colleagues note in a brilliant concluding thought from our sources, an hour in the library will save you a week at the laboratory bench.
An hour in the library saves a week in the lab. That is the perfect validation for putting in this intellectual work. You are building familiarity, establishing confidence, and turning what used to be a brain-freezing wall of text into a clear, highly navigable blueprint.
You're transforming from a passive victim of information overload into a highly capable, active investigator.
Exactly.
Which leaves me with one final thought for you to chew on. We've spent this whole time talking about applying this highly critical, nonlinear, meaning-making approach to complex scientific papers. But if this active investigator mindset-checking funding, separating raw data from the author's interpretation, and challenging your own biases is the best way to decode dense science, well, how might applying this exact same approach fundamentally change the way you consume everyday news articles, opinion pieces, or your daily social media feed tomorrow?